In this video, we will talk about diabetes. Let's start with a quick introduction. Diabetes is a chronic condition characterized by hyperglycemia or increased blood sugar levels. There are mainly two types of diabetes, type 1 and type 2. Type 1 diabetes is usually described as an autoimmune disease, and type 2 diabetes is generally the consequence of bad eating habits that lead to a resistance to insulin. In this video, we will focus only on type 1 diabetes. Before we dive into this condition, let us analyze what is an autoimmune disease. Autoimmunity is the response of the adaptive immune system to antigens expressed by the body's own cells and systems. Basically, and for many reasons we will discuss later, the T lymphocytes destroy the insulin-producing cells. These are called beta cells and are found in the pancreas. Their main function is to secrete insulin, which in turn travels to target tissues and acts as the key to a lock to let glucose inside the cells. So, when the T lymphocytes destroy the beta cells, no insulin is produced and glucose cannot enter the cells. Consequently, the concentration of glucose in the blood increases and the cells cannot perform their functions because the intracellular decreases. This destruction of the islet beta cells in type 1 diabetes is a result of a complex interplay between multiple players in both the innate and adaptive immune system. Now let's discuss the causes. Type 1 diabetes is a polygenetic disorder, but there are also non-genetic factors, most likely environmental, that are also involved in the pathogenesis of the disease. All this results in a T cell-mediated autoimmune attack against pancreatic beta cells. There are many genes that produce type 1 diabetes, but there is a specific one in a loci within the human leukocyte antigen HLA that accounts for most of the genetic susceptibility. Current evidence suggests that initiation of type 1 diabetes requires both CD4 and CD8 T helper cells. T cells can directly kill beta cells via cell-to-cell contact through cytotoxic processes, but they can also influence their destruction through other factors, including release of pro-inflammatory cytokines, granzyme B or perforin, and possibly signaling through pathways of programmed cell death. Now that we've discussed this introduction, let's talk about the pathogenesis of the disease. There are many things that influence the destruction of pancreatic beta cells. We're going to take them on one by one. Here we have a pancreatic beta cell, and we're going to see the different pathways that lead to its destruction. So let's start with the first one, that are the dendritic cells. Dendritic cells are an antigen-presenting cell that recognize antibodies with a receptor called MHC that we've talked about. Now, the presentation of the HLA gene that we talked about creates pancreatic islet autoantigen that are reactive against pancreatic beta cells. Dendritic cells recognize this autoantigen and then present it to other cells of the immune system. The presentation of these antigens would be step number 2, and dendritic cell presents them to T cells, both CD4 and CD8. When T CD4 cells are presented with this autoantigen, they recognize it and they start differentiating. This would be step number 3. After recognizing this pancreatic islet autoantigen, the T CD4 helper cells suffers a cascade of differentiation, and they turn into 4 different types of cells, actually. Th1, Th2, Th17, and the Treg cells. Each cell activates a cascade of cytotoxic effects that end up in the destruction of beta cells. Th1 produces interferon gamma that activates the STAT1 pathway for the destruction of beta cells. It also secretes IL-2 or interleukin-2, which helps activate the T-CD8 cells and the Treg cells. So here we see that with Th1 cells, there is an enhancement of this cascade by the STAT1 pathway and the further activation of CD8 cells and Treg cells. Th2 cells, on the other hand, secrete a different type of interleukin. That is IL-4 and IL-10. Now we know that these interleukins activate plasma cells, and plasma cells in turn produce antibodies. These antibodies are also directed to beta cells. So IL-4 and IL-10 activate the plasma cell, and the plasma cell secretes the antibodies, and these are targeted towards beta cells and their destruction. Now T17 cells produce IL-17, which are also part of this cytotoxic cascade that enhances the destruction of beta cells. And lastly, let's talk about Tregs. There is still research going on about these cells because we don't know exactly what their function is on this cascade, but we do know that they're activated as a consequence of the dendritic cell presentation to the T CD4 cells. All of these interleukins and all of this cascade is labeled as step number 4. Now let's talk about T CD8 cells. These secrete 3 different types of proteins: FAS, TNF alpha, and IFN gamma. All of these are also directed towards the pancreatic beta cell. Lastly, we have step number 5, which are the natural killer cells. These are involved in direct killing of beta cells through the interaction of NAK cell markers such as NKp46 and KRS. As we can see, all of these cells secrete their own proteins that end up in the destruction of pancreatic beta cells. All of these cascades take time in activating, but at the end of this, we have a very sad and destroyed pancreatic beta cell. So we have no production of insulin on this person. Now remember the pro-inflammatory and granzyme B and perforin that activated the HLA gene? These also have a destructive effect on the pancreatic beta cells. This last part ensures that the process is not centralized on a single beta cell, but actually a systemic effect that inherently destroys every pancreatic beta cell. At the end, we have no insulin-producing cells, and this patient becomes an insulin-dependent patient with type 1 diabetes characterized by hyperglycemia.